Yeah, thank you very much for this very kind introduction, and really, I'm really appreciating that I can be here and talk here. I mean, it's my second time to India, and I really love it. I mean, Bangalore is a fantastic place, and I've seen these wonderful, vibrant places like across the NCBS where Shashi is, who did his PhD with me in Göttingen, and I see all these other places. It's really just fantastic. CTS and IISc and all these places. So it's really an honor to be here, and I hope I can tell you something you might be interested in about model microswimmers. Actually, I should announce that our whole work on model microswimmers started off when Shashi did his PhD with me, because this is his invention. We were always interested in physics far from thermal equilibrium, pattern formation, collective behavior, and things like that. And then he came one day with this wonderful system where you just have an emulsion of oil droplets in water with just the right surfactant, and they swarm and they keep going. I mean, it's like starlings swarming, and you can see, well, how do they swarm? And they are a very nice model system to study the basics of swarming, because this is physics far from equilibrium. You cannot apply standard Stephan and all these things. And so this is a whole not yet finished, by far not yet finished research field. So of course, it's not just because it's fun. The reason we are dealing with these things is also very practical. I mean, if you look at photoactive microorganisms in nature, like also microswimmers, which is, in this case, which is just plankton. I just have to see, oh, this is one. Okay. Now this is, which is, yeah, maybe turn down the light a little bit. I don't know whether I can do this here. Okay. So what you see here is the top view of an algal bloom, and you see there is that the plankton in the waters is not evenly distributed. Actually, it's not even that it's a single species. There's striations of species, and it's not clear where they come from. And as you can see here, quite obviously, it couples to the streaming, to the streaming patterns in the waters. And the reason why this is really important is that that the plankton in the oceans, I mean, this system through which all of the solar energy is fed into the ocean system, to the whole biosystem. This is then the bottom of the food chain, and then it starts, from there it starts on by predation. So it's a really very important system in order to understand the energy coupling into the Earth system, into the biosphere. And so it's really something you want to understand from a very basic point of view. And here we are now dealing with these photoactive, because it's the photosphere. Yeah? The light comes in, they do photosynthesis, they put the energy, solar energy, into chemical energy, and this is where it all starts. But they're not only in the water swimming, they're also in completely different environments. This is, as far as you can even conceive conceptually from this is in the Antarctic. So if you go into the Antarctic, and break one of these of the rocks open over there. This is a sandstone, and you see this little hue here. This was the original surface of the rock. You see this hue here. These are algal organisms which live in the rock, in the interstitial spaces between the grains, because the environment here is much too hostile. You don't want to be there. But they manage to live here, and this little bit of light which comes through the grains in here is just enough for them to live. And I mean, they are the only creatures living there, except yourself, if you go there and break this off. I mean, it's completely hostile, but they're living in the rocks. So these microorganisms are very, very successful at adapting to different environments, and this is why, if you study them, you have to be prepared for studying quite a number of different geometries, different conditions, and well, that's what we are set out to do. It's not only for studying nature. These microorganisms are also very important for biotechnology. For instance, there are these power plants of algae. Yeah, you grow algae and then you harvest the the carbohydrates and get energy out of that, just biofuel. This is one way. And of course, they are also very interesting for biosynthesis of pharmaceutical drugs. So there are many reasons why it's interesting to study them, and there's a lot which is not understood. So our model organism, the first model, so I will have talk about the artificial ones, but also models. Some model organisms you can take which are very well described in nature, which you can just take out of the biosphere. So one very nice model organism is Chlamydomonas reinhardtii. It's a little cell like this. It has two flagellae, and it swims like this, like breaststrokes. And it is also nice for looking at some model interference between two different ways of dwelling in the environment, which I already alluded to. One is the cell suspension, which is the model for the planktonic state. This was the plankton in the limnic or in the pelagic environment, and then you have surface adhered state, which they also do. I mean, you see they do adhere to surfaces, and this is of course a state which will be very much dominant in environments where you have lots of surfaces, like in the Benthic, when they are on the seafloor, or even in the Antarctic when they are living in the interstitial spaces of rocks. And they can do all kinds of all these things, as we have seen. So actually, you don't have to go to the Antarctic. I mean, the natural habitat is really also soil, and as I said, the Benthic, so the ocean floor. So this is very, very natural also to navigate in between these habitats like this and the free habitat in the planktonic state. And one thing you might ask is, well, how can they possibly adapt to so different ways of dwelling? So what governs the transition between these two? So the surface adhered and the free swimming state. And this is, of course, about measuring forces between a bacterium or an algae cell and a surface. And in order to measure these forces, I mean, there are several force measurement devices out there. You can use optical tweezers or the atomic force microscope, but then you're typically here in the piconewton range, and if it comes way beyond piconewton, you get out of range of your device. Then, of course, you have dynamometers and force gauges. They're usually a micronewton to newton, something like that, but you want to be here. So you have to devise some method how you can just measure these nanonewton to micronewton forces of these cells. And this is how we are doing it. So we devised these micropipette force sensors. This is what Oliver Bäumchen has brought from his postdoc to the lab, and he has developed it there. So what you do is you have a micropipette. This is now very thin, so thin that you can aspirate a cell to it. And it is not a straight pipette, but it has a double bend. So it goes like this, and it has another bend over here. So you can bend it. And from the deflection, you can measure the force. And this actually works quite well, but the way you have to do it, you have this little micropipette here with this double bend. You suck the cell to the pipette, and then you can approach a surface under the microscope. So this is between glass slides, and you can approach it with the microscope. And here you see one of these cells doing its breaststrokes, just sucked to the pipette. And this is this micropipette where we want to do the measurement. And the nice thing here is we can do the visualization and the force measurement in situ through optical microscopy all at the same time. And we can do in vivo measurements really of motile cells and tissue, because as you can see, it doesn't harm it, and it just stays alive. Yeah. From the bending, I come to this in a second. Yeah. So this is how it works. So this is the pipette comes from here. This is the bend, comes over here and has this final bend, and here's the cell. And then if you make a cross section here and just look at the gray levels, you see it looks like this in the gray level. And if you correlate these two images, you just look at the maximum of the correlation between these two data sets, And this is very, very accurate. And you can easily resolve piconewton resolution. So we get a resolution of 10 piconewton force resolution. But of course, we can also measure rather large forces for large deflections. And this way we can very accurately measure what the interaction forces between this cell and an approached surface actually is. So, and this is what we saw. I mean, people have been doing zillions of experiments with clummies. And what I now show you, you would expect, I mean, this must be known for a long time, but actually it was discovered in Oliver Bäumchen's lab in my department, and was something which really struck us. What you do is, see here, is this clummy sucked to the pipette, and these are just, we enhanced it here. These, otherwise they're hard to see. These are these flagellae. And now, this is the deflection of the cantilever, and this is here by 0. Now, all we are doing is, we are illuminating here only with red light. So it's only red light illumination. And now we switch on blue light. And after some time, well, it's beating with its flagella, and after some time, it happens to touch the glass again, and then it sucks itself towards the glass with a force of a few nanonewton. We switch the blue light off again, going back to just red, and it lets go again. And you can keep going. I mean, this is switchable. You can switch on and off and on and off and off. So the adhesion between the cell and the substrate is switched by light. And it is only some spectral part of the light. If we did this with several different wavelengths, and you see you need 470 nanometers, 550 is already too long. So there's a threshold, and this corresponds to one photoreceptor, which is known for this, but there was no knowledge about a signal cascade which leads to the adhesion. This was entirely unknown, and you can so here you can see that it is completely reversible, and you have a sharp photon density threshold. If you need a certain threshold of photon density for this effect to kick in, and typical solar light is somewhere here. So at something like 30% of daylight, it realizes, oh, there's light, and I should stick. So at first glance, this seems completely odd. I mean, how should such a cell, why should it do that? But actually, at hindsight, it's very reasonable, because since it also dwells in the benthic granulate, if it just swims and keeps going and makes a random walk, it may be lost in the dark and never do photosynthesis again. So what it does, it makes a random walk until there's a little bit of light, and then it glues, and it stays there. And it's absolutely important for its survival, such that it's a very clear advantage in the survival, because it is doing photosynthesis. And yeah, okay, maybe I should. Okay, blue light, good question. Blue light, it's just, well, this is just the intensity or the sensitivity curve of this photoreceptor. Why it does not adapt to the red light, I don't know. Maybe it is because, I mean, you need a certain threshold photon energy to kick off the chemical cascade. Maybe red photons are just not strong enough, and so it does adapt to the blue photons, and the red don't interest it. This is possible. The discovery, of course, I mean, it is only because of this we could discover it, because for a completely different reason, because one camera had this sensitivity curve. Christian Kreis did an experiment with a red filter, and then suddenly the gluing didn't work anymore. And he said, well, what's going on? And so this was just completely accidental discovery. And what was also interesting, I mean, I asked, why has this not been discovered a long time ago? Because there's tons of literature about clummy. Well, the point is, it turns out the strain we are using, this is from the Göttingen algae collection. This is one of the 3 biggest algae collections in the world. This is sitting in Göttingen. And there are others. One is in Cambridge, one is in, I don't remember. There are several large ones. Most of the publications which are done with clummy and which are said allegedly wild type, they are from one of well, there's one, I forgot which one, of these other depositories. But the point is, all the clummies which have been investigated stem from one single cell which has been dug up in the middle of the last century from some potato field somewhere in Massachusetts. And this guy has then proliferated them, and on the way, one had metabolic damage. So there was some mistake in the protocol, and it got its nitrate metabolism was damaged. So it has no nitrate metabolism. It's fine, everything fine, but the gluing doesn't work, or at least this mechanism doesn't work. So it's not the wild type. It's just, it seems like the wild, but it's not. For the gluing, which is really evolutionarily important, it's not the wild type. So if you want to be very strict, you have to redo all these experiments, of course. It's not that bad, but one should be aware that it's not the wild type. There's the real wild type. If you go into a puddle and dig up another clummy, it will do that. And we did that. I mean, we did it from a Japanese depository, which have completely independent strains, and it also does this effect. So it's the damaged ones which don't. Okay, but now, as we said, this is important for the various habitats. So you have free habitats, and it glues to surfaces. Surfaces are there where you have small geometry, then surfaces become important. So it is, of course, not only important to look at the gluing to the surfaces or at the interaction with the surfaces, but also, are there some peculiarities of the behavior if you come up with confining geometry? Now what you see here is a clummy in a microfluidic micro Petri dish. So this is 100 microns across. And you could see, well, it looks like it likes the wall. I mean, it's somehow, you have the feeling it's not all over the place. It rather likes the wall. So we looked at this in some more detail, and you see, and we varied the diameter of the Petri dish. So if you have 0.5 millimeter, you see, well, this is the heat map of the frequency. You see, it's a little enhanced close to the wall, it looks like. If you make this dish smaller and smaller, you get a very strong enhancement. This distance here is just the half diameter of the clummy. So this is just depleted by hardcore repulsion. So if you look at the radial distribution, you see that as you make the Petri dish smaller and smaller, this enhanced probability near the wall gets larger and larger. And now you can try to simulate that. I mean, one want to see what is the reason for that. The way we did that was we made a model where the clummy consists of one sphere, which is just the body, and another sphere, which is somehow the range where these breaststrokes happen. And and we have a force dipole. We put two Stokeslets here. We have a force dipole which takes care of the propulsion. Yeah, but it bounces off. The point is that it turns that if you have this geometry, what comes out in the simulation is that it bounces off at a preferred angle, at a preferred glancing angle, and this has already also been seen in Ray Goldstein's lab. If you look at the scattering of these particles from straight walls, and the result is that it tends to do some kind of a polygonal move, and this gives rise to the enhanced probability close to the wall.
Particular organism, but if I just thought of an active particle which will move in a given direction with some diffusion in the direction, like the usual kind of generic active particle model, I would expect that it would show enhanced density at the boundaries anyway, right? So how do you distinguish that from this?
Well, I mean, if you make the distribution of angles by which it comes off the wall completely featureless, then you do not get any enhancement. Yeah. Then it's really homogeneous. You need this preferred smaller angles. Otherwise, it doesn't work, really. So, I mean, we really have to put in the characteristics of our clammy creature into the simulation. What we do actually is some kind of a Langevin equation simulation, and then we get in the simulation we get quite just the same kind of enhancement as we get in the experiment. And if you then plot the height of this peak or the area under this peak as a function of the radius of the petri dish, then you see it goes down like this.
Yeah. No, sorry if I may. No, I mean, if I imagine a particle that's moving persistently towards the wall, so it hits the wall and then it can't move forward, the only way it can escape is through a reorientation of the...
Oh, okay.
Right?
I see.
And then generic, I'm saying, then it will sort of have a preferred angle of coming out, which will be a very small angle with respect to the wall, because that's when you get free.
Okay, so if the rotational diffusion is very slow and it takes off as soon as it somehow gets the right angle, yeah, so this would be maybe one rationale why the angle distribution is not featureless. Yeah. Actually, if you look at the motion, the rotational motion is very fast. So I'm not sure that this is really the mechanism. It may be just geometry that the breaststrokes are larger than the body, and so it goes preferred at some distance. I mean, we will see that there are also hydrodynamic interactions which just come from this pumping of water. This is what we see then only in our model microswimmers. And there the effect is even stronger. So one sees that one doesn't need that whole mechanism. There are also hydrodynamic effects which do that. Yeah, let me just go on. So the question is can you just describe it as a function of the curvature? And it turns out, yeah, you can. I mean, you can just put them into an elliptical chamber, go away from this trivial circular one, and you see the enhancement is the stronger, the larger the radius of the local radius of curvature is. And then if you compare our experimental values with the simulation, you also see here, well, it's just the local. Here it's just the local curvature. The local curvature governs the enhancement. Why are we studying the enhancement close to a wall? Because the enhancement of a cell density close to a wall is a precursor to a biofilm formation. This is why it is also very important to study that. Because biofilms are, if I go back to these bioreactor things, biofilms are what really mars the whole project. You get biofilms in these reactors and you have to continuously clean these tubes. And this makes it, yeah, not doable, more or less. I mean, this is really the biggest problem. So if we can control the biofilm formation and control the gluing to a surface, then we can maybe do bioreactors which are much more efficient. So this is also why it is very important for applications. Yeah? Well, I don't know, but it could be, yeah. I mean, biofilm formation is a very, very complex process, and a biofilm usually does not only consist of one species. It's a whole city, yeah, with different species, and it has a stratigraphic differentiation and things like that. So this is a very complex thing. I wouldn't use that simple picture. So the next thing, now I come to Shashi's invented bugs, to do something which is a little more simple. As you see, these are very complicated creatures. They have these breaststrokes. They have, of course, a life cycle, and you see they do adhere. And well, it's hard. Of course, you can come up with hypotheses how biofilm formation comes up, but you want to have something really simple, which does similar things. So here is our model system. What we do is we put in oil droplets in water, where we also have a surfactant, in a formulation where the oil wants to form a microemulsion. So the oil wants to go into, so the minimum of the free energy is a little bit of oil in each of these micelles. You have empty micelles here, you have filled micelles here, and the drop in free energy from this state to this state provides some energy, and it turns out, and I hope this, now the movies come, I hope this is a problem, maybe I can make it run here. This doesn't work. So I hope the other movies go. So what you are supposed to see here is just a droplet in water, and you see it moves ballistically, and it leaves some kind of Schlieren behind it. What happens is that the way this process proceeds of the dissolution of the oil into the micelles is as it is depicted here. So if you happen to have some willowing motion in the droplet by some fluctuation, so you have some flow along the surface, then you advect empty micelles from here, and as oil goes into the micelles, you need some, because the micelle swells, you need some extra surfactant, and this surfactant can come from nowhere than from this surface. So along the way, it eats up surfactant, which means that the surface is surfactant depleted over here. But a surfactant depleted surface has a higher surface tension. So you get a Marangoni stress in this direction, so the Marangoni stress sustains the flow. So this gives rise to a runaway instability, which makes the droplet keep going. That's the way how it works. So, now, come on. So now we go back to the geometry. Now we have our droplets, they are free running, and now we provide them here graphically with these traces so you can follow them. Here we have, in a microfluidic device, we have now put pillars. So this is the opposite. It's not a petri dish, a micro petri dish, is a micro pillar. Oh, come on. I do that again. Now. So you can see, they also glue to this surface even though the curvature is negative. So it has a very strong propensity to stick to the surface. And here we do not have these tentacles. It's just hydrodynamics. Now there's a question. Can I say that? It's a pillar. It's a pillar. So here you have okay. Let's do that again. No. No, I want you to. Now, okay. So these are two pillars, and around each one there's one droplet which just goes around. And you see that it very strongly likes the wall. And really just due to hydrodynamics. If you make the pillars smaller, other things happen, as you see here. In principle, you have the same thing. Goes through the pillar, goes around, but then it smells its own exhaust. It doesn't like that, so it goes off. So you have two things which you have a trade-off. It wants the wall, but it also doesn't like all this debris. So there's some chemotaxis which takes it off. And if you make the pillars even smaller, you see there's almost no propensity to stay at the pillar anymore. It goes around a little bit, but it doesn't really like that. So we again have a curvature dependence, but it's somehow the other way around, of course, because the curvature is negative here. Now I have to see how I can. Yeah. Oh, sorry. Size of the drawing, roughly 50 microns. Yeah, I mean, it varies. It doesn't depend strongly. Of course, the droplets get smaller over time because they are dissolved in the oil. Yeah. And if you say, well, they don't like the own exhaust, what is so particular with the own exhaust? It's a depletion of empty micelles, right? So you could imagine that they go for more empty micelles, and that's what they really do. If you put a droplet with a concentrated surfactant solution over here, these are the traces, and they really go for this droplet. I mean, they're very strongly attracted, so they have also chemotaxis here. And you can see that also their interaction with each other has a lot to do with these exhausts. You see, they don't cross. Yeah? So you have a strong interaction which is governed by this. It's like a repulsion. It doesn't like the trail, like the trail. Yeah. There is not so much of. So I'll show this in a minute. So I mean, here you can see it right away. What happens now? This is a channel which. Nah, sorry. Okay, maybe before I started, I explained. So this is a microfluidic channel. These are the droplets, and you have these like little islands where they can go either there or there. And you will see as the movie plays that if one droplet went this way, the next droplet will go this way because it tries to avoid the exhaust of the other droplet. This one. Okay. So here. This goes up, this goes down. This goes up, down, this goes up. Yeah? Up, down. And you can see if you increase the delay between the droplets, it decays because after time the stuff diffuses and then the memory is gone. And here in this movie, what we did is we put some Nile red into the oil. This is actually the oil. It's not important here, but I just say it already because it will be important in a minute. As the oil, we take 5Cb, we can take a liquid crystal, which is also very interesting for looking at the direction of its motion. Here we put some Nile red in, and you see that the dye gets into the micelles along with the oil. So we can see where the oil really is, where the filled micelles really are. Okay. Okay. Now here. So you really see that this is the exhaust of another droplet, and as this one comes here, as soon as it sees that, it bends off. Okay. So it's really the depletion. Well, it's the filling of the micelles. They are not useful anymore for the propulsion, so it goes away. And this means that if you have many droplets, now it's for the first time I want to go into the collective phenomena. If we have many of those, they're seeing their own exhaust, but also the exhaust of the others. Unfortunately, this is a jerky, but we have traced a few here. If you look at the traces as they develop, well, we have to wait for a little bit. I guess it's going to stay jerky. Maybe you look at, yeah. So you see here that they start off quite ballistically, but after some time they start to curl very strongly because they cannot go anywhere anymore. Yeah, it's like a cage effect. They're stuck there, and the nice thing here with this system is you have, if you have a dense colloidal suspension, you have this cage effect, so you cannot go anywhere because of the other colloidal particles. And mode coupling theory is one that uses this cage effect as a paradigm. So you have this memory effect in which then does away with the possibility to move. And here the system is nice because we have a cage effect which diffuses out over time. So we know we have a memory kernel we have a memory kernel in the system which decays over time exponentially, which makes the math very simple. So maybe the collective freezing of the system can even be very rapidly treated with mode coupling theory. We don't know this yet, but this is one project we want to follow. So, as I already told you, it's not a simple oil. We use a liquid crystal, and you see it has also some structure. Here you see the traces are completely curled. We run this again. Okay. The traces are curled, And the question is, what happens here? And is it always that way? The answer is no, it is not always that way. In the liquid crystal, the nice thing is we can by tweaking the temperature a little bit, we can switch between isotropic and nematic states. And here, we just ramp the temperature, so the droplets are making this curled motion, and as soon as we get to the isotropic state, they go on ballistically. So the curling motion has to do with the internal structure, with the nematic structure of the droplet. And we can also see this, of course, in correlation function. If it's isotropic, the angle correlation just goes down exponentially because it's like a random track, it's like a worm-like chain, so it forgets its direction, whereas here, of course, you have the simulation due to these curls. So what happens here? Well, if you look at the droplet locally, this is a nice thing about the liquid crystal. You can look at it under cross-polarized, and you see there is a defect sitting, and the defect is somehow advected by the flow field to a certain point, and the defect is at one point. You don't see it here, this is only schematically, but you can see it in the microscope that you can see a defect, and you see where it goes, and usually you find there is a certain angle. And now if you take the droplet trajectory and you plot in the same plot, you can plot the direction of the defect vector, you see they have some angle with respect to each other, and of course if they also cross, if this droplet crosses its own exhaust and it's not so strong that it still can't cross it, then of course this angle is somehow affected, and then accordingly the radius of curvature changes, then you get this curly motion. So it has directly to do with the nematic director structure, and this is what makes it also a nice playground, because you can incorporate in a controlled way all kinds of well, interesting behavior. And if you go on in this direction, I mean, what you really want is to go more towards a cell model. Now we have an oil droplet in water. Cells are, of course, vesicles. So you have water, and then you have a shell, and then you have water again. If you want to go closer to life, you want to have these double these shell structures, and we can make them. So what we have here is we have water droplets in oil, and we put them again in, no, sorry. This is water, so the white is water, this is oil. So we have water in oil over here, and we put it back in water again. So we have here shells of the dark phase is oil, liquid crystal here, so this is the liquid crystal phase. We have water droplets in these liquid crystal droplets. So we have these shells, and if we look what they are doing, they are also going. So it is enough to have this little oily shell here. This does enough of this dissolution, and it also does the Marangoni instability. They're just a little more bizarre in their way they move, but they do. And actually what we found quite amazing is that these shells grow very, very thin, because before they finally collapse into just a little, very tiny oil droplet. But they really grow almost vesicles, and it really works. And so they're very nice for some model cells, and for some even having some cargo in. And we can look, if we put some colloidal particles in, we can even look at the velocity field within the aqueous droplets within the oil field. And yeah, they do very similar things than those we have seen in these nematic droplets. Of course, here again, the director field interfaces with the position of this droplet. It's a whole system which adjusts its geometry accordingly. We can also have, now that we say we go towards artificial cells, so we have now vesicles. Of course, the next thing for a proliferome would be the cells have to be able to split. And well, I mean, you can do similar things like that here, because if you now have these droplets and squish them between two plates, so you go to two-dimensional systems, you can show that in principle, it's not only in this flow field around the droplets, not only the first Legendre polynomial which can kick in, but it's also higher Legendre polynomials, as you can see here. Here you have quite a number of these lobes, and you can see here how the droplet moves. You have quite a number of these lobes of flow around. Here in this artist's view, it's a little more closely, more clearly shown. And we have also observed that if you have enough, they do rip the drop apart. So you can even split the droplets. So you could cook up some ideas of motility and growth and proliferation by splitting. Here we have, we made it visible by some PIV. So here you have just the second Legendre polynomial, which has become unstable. So you have a more complex flow field, which is all driven by these Marangoni flows. The Marangoni flows are so strong and can give rise to all that, because if you consider the Marangoni velocity, this is some of the prefactor of all that. The Marangoni velocity, this is a typical surface tension divided by the viscosity, and for water, this is around 70 meters per second. So if you're here talking about micrometers per second, something like that, I mean, you need only something like 10 to the minus 5-ish imbalance in surfactant, and is already enough to give rise to rather rapid transport. So this is why Marangoni effects are so strong in these systems, and you have plenty to do not only the first Legendre polynomial, but also all the higher ones. Now, let's go deeper into collective behavior. I mean, there has been a lot of talk about collective behavior far from thermal equilibrium in these swimmers, and there have been many theoretical papers about motility-induced phase separation called MIPS, yeah, where they show that just motility already should induce some kind of phase separation or some kind of clustering, from the simple idea that if you have ballistically moving particles and you have accidentally a cloud where they are more dense, they run into this cloud, they bump into the other particles, the ballistic motion is stalled, and so they don't get away anymore, and you have even more clustering. So this is the idea behind it. It has been simulated and it works. The problem is, it is difficult to observe in such a system, in a real experimental system. And the question, somehow, it doesn't work so readily as as on the computer. And here, it's very, very dominant, very, very clear. And so you would say, okay, no, finally, we have motility-induced phase separation. The problem is, it is not. This has nothing to do with what people have simulated on the computer, because what is important here is buoyancy. If we take this and the aqueous, well, the liquid crystal is more buoyant, is heavier than the water. Now, if we add D2O, such that at a certain point we have buoyancy matching, the effect goes away. If we have too much D2O, then they are lighter. It's such that we have again a density difference, then it comes up again. So very clearly, it must have to do with the coupling between the droplets and the water. And this is what I said in the beginning. Swimming coupled to the flow field of the liquid is really important also in the limnic and in the pelagic environments. And it is here, even in the Petri dish. So we can prove that it has to do with it. It's very simple to show and to prove what the length scale then really is. It's very simple. It's convection rolls. So we have these clusters, and they collectively pump the liquid, and they create convection rolls. You can prove that by changing the height of the Petri dish, and then you indeed find if you increase the height, also the wavelength increases. It's just convection rolls. It has nothing to do with an intrinsic length scale of some Cahn-Hilliard thing in the motility. It's really the coupling to the hydrodynamics. So hydrodynamic coupling is really important in these systems, and usually in most of the simulation sets has been overlooked so far, at least in the beginning. This again is to remind you how important it is. This is the Falkland Islands. This is latitude, longitude. I mean, this is a large area in the ocean, And the fact that you have, I mean, it's a color code, so each color is a different species, and the fact that there are so many different species and they come in striations is one of the things which is not understood. And in order to understand that, we also have to understand the hydrodynamic coupling, also buoyancy coupling, to the streamings in the photosphere, in the pelagic zone. So let's go a little bit more deeply into that. I mean, if you have hydrodynamic coupling, it turns out that, well, you can also simulate this and see if you simulate also the liquid around it, not only the swimmers, but also the liquid around it, then you can look what do they do on the computer, and you find not only just motility-induced phase separation, but you find that as you increase the filling fraction with the swimmers, the clustering goes up and goes down again. So what do I plot here? This is the width of the distribution of the local density divided by the width of the random distribution you would have. So it's something like giant number fluctuations. Yeah? If you have clustering, then if you do local probes, then you get very strong variations in density. These go up, so this is clustering, indicative of clustering. They go up, but then there's a maximum. And if you make it even denser, clustering goes down again. So this is clearly not this typical signature of motility-induced phase separation. There's something different which kicks in. And the nice thing about these systems is if you approach them in this way, you can even write down a Fokker-Planck equation in the position direction space, and you can do a linear stability analysis of the whole thing. You can do a linear stability analysis of this density distribution, and what you find is that the hydrodynamic terms destabilize. These are the yellow terms, both for pullers and for pushers, and the hardcore interaction, so the steric terms, they stabilize. So what you get in the end is, well, you can now, here you can separate these terms. You feel that the clustering comes from the hydrodynamic interaction. And it's the steric effects which do nothing like that. The steric effects is actually what stabilizes against the instability. So this was, but the steric effects was behind this motility-induced phase separation idea. Yeah, the ballistic ones bump into, by steric effects, they bump into the others, and so they thought it's just not true. These are stabilizing it. Yep. Oh no, sorry. They stabilize with respect to the clustering instability. So clustering I consider as the instability. So you have a fluctuation and it becomes unstable, which means that it clusters. Clusters get even more clustered, depleted areas get even more depleted. This is unstable, and steric stabilizes. So steric tends to smoothen it. It's the hydrodynamics which makes it unstable, which is counterintuitive, but this is what comes out even in the analytic treatment. Yeah?
Okay, so I was trying to understand this as, there is MIBs which would come from the activity, but if you add hydrodynamics, there are additional features that emerge.
If you add hydrodynamics,
that
actually you get a completely different system.
Okay.
Yeah.
Okay.
It's a completely different system. If you just consider them as running ballistically in vacuum,
right.
It's just not the natural system. There is no such system, and it behaves differently. I'm not saying they're doing wrong science. They're not. I mean,
but it's space physics.
It's just not there. Yeah, it's just not there. Yeah. Yeah. Pardon? Steric is just that you cannot put two particles over each other. Yeah, so just hardcore repulsion. So now let's finally at the end go back to these natural swimmers, because then now that we're starting to have fun with the collective effects, I mean this is also collective, right? So we should see what are these natural swimmers doing collectively, and there really the puzzles come up. I mean this is again Clammy, and I've showed you that if you have red and blue light, they completely change their behavior as far as adhesion is concerned. What you see here is, this is clearly clustering. This is a 4 millimeter micro petri dish, and you see clearly, you see clustering here. So this is some collective clustering of the Clammies, but this only works in red light. If you shine in green light, they immediately spread out homogeneously. And if you look at it in some more detail, what you find is that this reproducibly builds up. If you have blue light, it's all, sorry, the color code is the wrong way. So if you have blue light, the density is completely homogeneous. If you switch to red light, it gradually, over time, this builds up and is completely switchable and reproducible. And if you look more closely, if you increase the magnification, what you then see is that in the dense region, they are slow. So this is just multiple exposures such that each particle effectively gets a tail, with the length of which is proportional to its velocity. So here you see all these long tails. So here they are fast, here they are slow. So it's like in MIPS. The denser they are, the slower they are. But what is completely enigmatic, and we have no explanation whatsoever so far to that, although we had several checks, I mean, it's not that here you have a wall and here you don't have a wall. It's all over walls. You have walls all over. I mean, this is a Herrlich-Hall cell. So there are sea walls everywhere. I mean, there's a bottom wall and a top wall. There's this additional wall over here, and this seems to make the difference. And this is something we don't understand. So we are doing several experiments, also changing the materials. We did that, no change. It's very robust. We did several geometries, and it's very robust, and it's light switchable, but we have no idea what it is so far. Okay. Well, we haven't tried so many. It's not so easy to completely vary it, because you have to change the formulation of your microfluidics. But we're looking into that. I mean, this is really something I honestly must say, we don't understand it. It's just even also collective behaviors. Yeah, there are surprises in the natural ones. And if you go to other algae, I mean, this is just only one algae. You have only these spherical ones, but there are also others, also in the benthic and the pelagic range, which are filamentous. And if you put filamentous algae in a Herrlich-Hall cell, this is about 300 fast motion. You see, I mean, they make really beautiful structures. And also like a liquid crystal, you have a -1 defect here, +1 defect here, something like that. And you can look at how these defects evolved. And this is now done by Stephan Herminghaus, who, sorry. No, I insist to do that again. You won't fool me. So this is now done by Stephan Herminghaus, who quite lately joined the lab, who came from Twente, from the Lose group, who's now also pursuing these more, geometrically peculiar algae. And now that I'm starting to talk about phases, this means I'm at the end. I will also talk about the other phases. All the Clammy group, the Clammy things were done in Oliver Bäumchen's group, who's with my lab. They have done all these adhesion experiments with the micropipettes. And then, of course, the theory is done by Marco Mazza's group. Marco now moved to a professorship at Loughborough, so I have to see who's going to continue with the theory. And then there was Corinna Maas' group. Corinna is back here. her group is pursuing all these wonderful artificial microswimmers Shashi has invented. Yeah, and that's it. Thank you for your attention. Oh.
So you showed like a couple of slides before that the clustering is happening more at the center. So does it depend on some volume fraction, like how many particles are there? Depends on some fraction that at lesser fraction the clustering will be more towards side walls or something?
You mean in this where the convection rules are kicking in?
Yeah, a couple of slides before you only showed like the clustering more at the center region.
The clump?
Yeah.
We have, I don't know. I don't know. I mean, there are, if you change the geometry, there doesn't seem to be really a straightforward way to predict precisely what the shape of the cluster will be. What you see is they try to stay away from the wall, from the vertical wall, for whatever reason. I mean, it cannot be gravitaxis. It's just too shallow. We don't know what it is. The main thing is you could anticipate most of the patterns by more or less saying, well, they're trying to stay away from the wall.
What about droplets case? What about droplets case? Will it be true in droplets case also when you are taking drop in some water or other oil, like other medium?
Yeah.
The clustering at the center.
No, they don't. They don't. I mean, the droplets are not light dependent. Yeah. The droplets are just what I showed you in this little micro petri dish. They make this Cairn-Hilliard type pattern, but it's all over the place.
In Zvonimir Dojek's lab with the microtubules and motors, do you know about this? Where they see flow depending on the aspect ratio. It doesn't depend on anything else. And I don't think they understand it either. But it seems to be something that depends only on the aspect ratio. And the last thing that you showed from Karpitska seems very similar to the kinds of defects, etc.
This is certainly akin to the microtubule thing.
Right. But in the microtubules in 3 dimensions, they also see this flow generated only when the aspect ratio is within some range. Otherwise, it doesn't flow.
Oh, I was not.
Which is that is so it's I don't think they understand it either. Okay. But there is certainly this.
That's interesting. Yeah.
Yes.
Thank you. Yeah.
So how does this adhesion of algae scales with the intensity of blue light?
How does the
Adhesion of algae, yeah. So you are using 470 nanometer where it is showing adhesion, right? So how does changes with the intensity at 470 nanometer?
Oh, it is a threshold. Let's see. So this is just the adhesion force as a function of irradiance.
Okay.
There is none until you get close to 10 to the 19 photons per square centimeter.
Okay.
It goes up.
And like, what is the maximum intensity one can sort of like it?
Well, if you go well beyond sunlight, they don't feel very well. I mean.
Okay.
Yeah, I had a question. This experiment you did for Chlamydomonas, which is basically a puller. So in nature, there are pushers or other kind of microorganisms also. So do they sense the same way?
Okay. Well, I mean, we didn't look into all these. I mean, it's physics. We are physics, but this is biology. You have this one organism you know how to cultivate. And of course, you are right, but we should do this for a range of different organisms. What we are doing now is we're trying to go away from this strain. So we teamed up with the biology group, with a group of Hegemann in Berlin, and they can do, by CRISPR-Cas, they can do all kinds of knockout and put other genes in, and we can modify this organism. So we can modify the metabolism, the gluing, and all that without changing the geometry of this thing. Because if we change everything at a time, I think we wouldn't learn very much. But of course, it keeps being pullers. It's all pullers. That's right. Yeah, we cannot change that because this is pullers.
A lot of work on the flagellar synchronization of clammies. Now, do you see any evidence in your collective behavior effects, any evidence or any role of synchronization as such?
We have not looked into that. It's difficult actually to look to see the synchronization of the flagellae and at the same time look at the collective behavior because it's a completely different length scale. You have to really go at high resolution microscopy to see these flagellae to synchronize and desynchronize. We haven't looked at it. I don't know whether it's possible to do that.
It might be possible to put in the simulation in some way.
This is true. This is true. We could, I mean, it's very well known, the mechanism, and it's also well known how to simulate it, so we could simulate it. This is true, yeah. This is true, okay. Yeah.
The clammies. So people have put bacteria into liquid crystals and then you can just visualize the flagella by the D.
This is true, but still they are on a small length scale. And they look in order to look at collective phenomena, you have to be larger as compared to that.
Yeah. So this is easily, so this is what Igor Aranson, etc. did with
right.
E. coli. So they've looked, so you can actually see the flagellar motions because of the texture of the liquid.
Yeah, this is true. Okay.
Possibly, I don't know.
I don't know how
well they live and so on.
Mummies digest the the liquid crystal.
I don't know. So you
We'll try. Yeah.
Maybe I missed it during the presentation, but I have a question about the fact that we can say that it's not the clustering is not due to the motility. During that experiments, it has been possible to vary the motility to exclude these things. I mean, it's...
Okay, I should be clear about that. I'm not saying it's not due to the motility. I'm saying it's not due to this standard idea of motility-induced phase separation, where you say it's just because they bump into the cluster and this is why the cluster gets larger.
So it's not because there is a threshold in the motility and in the pack, I mean, in the density. So as...
Yeah, I mean,
there are lots of...
It is due to the motility in the sense that you need the motility.
You need the motility
in order to pump these by pumping sustain these convection rods. So you need the motility. Without the motility, it doesn't.
Yeah, of course, but it's not. There is not a critical value of the motility given a certain density that gives you the.
Okay.
I mean,
yeah. I mean, what we are doing is we are doing experiments by varying also the viscosity. This varies velocity.
So
I mean, what we're doing is throwing this roll in. There you can vary the viscosity of the, but you also qualitatively change the behavior of the swimming. So I'm not sure it is easily possible to only
tune just the motility. Maybe with that argument there, maybe, I don't know.
Yeah, this is difficult.
more questions? Okay, if not, let's thank Stephan very much.